Hi everyone, and welcome back to this series of short videos about CytoPipeline, an R tool for building and visualizing pre-processing pipeline for flow cytometry data. In this video, we are going to start with the part I find the most interesting, the visualization of our results. And this is going to be done through a Shiny application. And so let's come back to our code, where we have just executed our pipeline by first creating a CytoPipeline object using JSON file as an input, then executing it using the execute statement, and checking the results in terms of the workflow of the different steps by calling the plot CytoPipeline processing queue function and having a check using this graph on the right hand side. Now time to visualize the results, and we do this by calling the CytoPipeline check app function from the CytoPipeline GUI package. For this function, we have one parameter which basically indicates the directory in which we have stored all our results. So let's launch this statement. So what it does is basically opening a Shiny application. So on this Shiny app, we have a two-sided layout on the first tab, so the experiments tab, where we have on the left-hand side, the user can select the sample and experiment, so the pipeline run he or she wants to visualize, and on the right-hand side, another pipeline experiment for comparison. So here, what we will do is the simplest use case in which we will compare the state of the data, so the flow frame, between different successive steps of the same pipeline run, so the same experiment. So we will select the same experiment name both on the left-hand side and the right-hand side, HBVT demo, which refers to the experiment name we have used in our code previously. And then the processing queue that we will have a look at is the pre-processing one. And we still need to select which sample we want to visualize, and we have two samples here, and let's select the first one. So by default, the same sample will be used on the right hand side. So by doing this, we have access to the current state of the workflow, which is all green and which is exactly the same picture as what we had before using the code. So here, what we will do is flagging the name of the object that we can see, that we can read in the green bubbles. So flow frame read object, for example, is a flow frame object which has been produced as a result of the flow frame read step. Same thing for the remove margin step, which produce remove underscore margins underscore object of flow frame type. So assuming we want to check what happens in these steps when we want to remove the margins, so in fact removing the outliers that are outside the range of the instruments, we can do that by visualizing it on the flow frames compare tab, where we select on the left hand side the before state, so the remove margins option, and on the right hand side the after state, let's say, so the remove margins option. So here that's the flow frame read option, and we want to see what happens after the remove margin. So let's select a 2D visualization, FSC, SSC. And so here what we see is that on the left hand side, so prior to running the remove margin, we have an x-axis which spans very, very high values, while after this remove margin step, the x-axis is now in the usual range of between 0, let's say small negative values, and a bit more than 200,000 as a value. Same for SSCA. So what is also very handy, apart from this visualization, is the visualization of the comparison, the explicit comparison, where basically the events that were present on the left hand side and now present on the right hand side have been flagged, highlighted in red. So this is a plotly visualization, so go plot. So what we can do is using the interactive plotly functionality, so we can see interactively the coordinates of the different points that we select. You can also zoom in using the mouse and so on. So this is Plotly, which can be handy if we want to dig into the values and look at the values that are read, for example. Okay, so this step seems to do its job correctly, because basically we tend to eliminate the values that are far out, so outside the range of the instrument. So let's move on to the next step, and when we do this, we will look at the remove doublet step, so looking what happens between the remove margins obj state and the remove doublet state. And we will also select the appropriate channels to visualize this step. So for the doublets, we know that we work in the FSCA, FSCH dimensions. So we will select those two channels for the visualization. Okay. And so for those who have visualized the introductory video, giving the context, explaining what it means to automate some of these steps, the remove doublet was presented as an example. And so there we discuss in details what was the principle behind this step. So let's just notice that indeed in this step we have removed all the doublets, so doublets for which the area is too high compared to the height, so and keeping only the events that are concentrated around the straight line here in the diagonal. Let's note that for each those comparison we can compare number of events before and the number of events after the step, and also have access to the percentage of removed events. Okay, so far so good. Let's have a look at the next step, which is, so if you look at the workflow graph, it's between remove doublets and remove debris obj. So let's have a look at that. Select remove doublets obj, remove debris obj, and then for this we will select the SSCA. Okay, so here the goal is to remove the debris which are concentrated as a cluster near the origin. We can see that indeed the step has done its job, at least partly, by removing this. However, it seems that those events that would like to eliminate as well, we want to concentrate on those events, are still present, and this is a bit rare, right? So I wonder what problem it might be, and so looking again at the JSON file, this corresponds to this particular step, which is remove debris, which we call the flow cluster package, and for which we have selected the parameters that seemed appropriate for this transformation. Do you have any clue what could have happened? Have you been able to find the answer to our problem? Yes? We'll check this in the next video, where we will focus on comparing two pipeline runs with different parameters on the same data.